All right, good morning. Last year as well, I had this first talk. Well, not the first talk. It was a panel discussion, but the first one in the day. So I don't know what it is with me and AppsCon. But yeah, it's good to wake up early and get the day started. So I'll talk a little bit about the work that we do in our lab at IIT Bombay. There are two main themes which are aligned with the focus of this session in our group, which is environmental monitoring and infrastructure monitoring. And what I would like to show you through some of the examples of our work is you don't really need to choose between low cost or high performance. You can really have both in the same platform. So I'll just try to illustrate how we have tried to do some of that in our work. So this is a broad overview of the kind of things that we do. So on the left-hand side, I've shown a kind of collation of all the different activities that we do for environmental monitoring. So the theme of interest in our group is wastewater surveillance. So we develop sensor technologies, sample processing technologies for looking for pathogens in wastewater. So this work started in our group in COVID-19. So COVID-19, there was a lot of push to look for tracking the spread of disease through testing of wastewater instead of testing individuals in a large community. So I'll talk a little bit about that. On the right, I've shown some of our work related to infrastructure monitoring. The picture on the top is a cover meter, which we have recently spun off as a startup. So I'll talk a little bit about that towards the end. But I'll talk most about the image that you see in the middle on the right, which is a smart cathodic protection system. So I'll talk more about that in a little bit. We also do some work on non-concrete structures. So on the bottom, I've shown a photograph of the ultrasonic inspection system that we have made, which was tested at IOCL. We had a demo of this and also some of the work on our wastewater surveillance yesterday. But I'll not talk more about that in the interest of time. Okay, so let me start with the environmental monitoring first, and then I'll get to the infrastructure monitoring. So before talking about what we work on, let's just take a minute to understand why is this wastewater epidemiology important and why or what exactly are we trying to do there. Like I said, this we started in COVID-19. So in COVID-19, I think this idea has taken a lot of momentum. There's a lot of people working on this now. But it's an old technique. It has been around for a long time. India itself has been polio free now for many years. And since the early 2000s, India, ICMR has an active wastewater surveillance program just to make sure there are no polio outbreaks. So the idea is that instead of collecting samples from individuals and then testing them in the lab, you collect wastewater samples. So these are sewage samples from a community. And most infectious diseases, certainly polio and also COVID-19, those who are, so if a community has somebody who's diseased, then the diseased individual, so if they're in a kind of state of the disease where they'll be contagious, they'll also start shedding the virus through waste. And then you can look for the signature of the virus in the wastewater. So the sample, so instead of collecting thousands of sample from a locality, you can just collect one wastewater sample and get an indicative idea of is the disease spreading in that area or not. The other advantage of wastewater sampling is that it gives you an idea of the outbreak much before hospitalizations increase, which is why it was interesting during COVID-19. In COVID-19, it was almost 12 to 13 days. So you have a lead time of at least 2 weeks before you start seeing large increase in hospitalization. So that's why it's called an epidemiology or a surveillance program, because you can use this as an early warning sign and also use this as a resource efficient way of doing testing. It's also a very great tool to do a lot of interesting studies. So I'm just showing here one of the papers. There are many, many papers that talk about all kinds of interesting studies. This was a paper where they looked at wastewater collected from long distance flights. And extracted nucleic acid from all the pathogens or all the organisms that are in the wastewater, and then did gene sequencing to come up with a profile of antimicrobial resistance, like a global profile of antimicrobial resistance, right? Because you have passengers from many different countries traveling on these flights. So if they shed their waste, then from that waste, maybe you get an idea of this. So again, so one is, of course, this is resource efficient, but also it's a wealth of information. There's a lot of things you can learn from wastewater. There's also a Netflix documentary on wastewater and all the kind of things you can do with wastewater. So if you have not seen it, please go check it out. So then the question is, I mean, what remains to be done? Because there's already so much that has been done on this. So we have to understand how this wastewater processing works. So the smaller image, I'll have a bigger picture in the next slide, but just to give you an idea, there's a lot of steps that have to be done starting from sample collection until you do analysis, which require a lot of expensive instruments and a lab. So that requires access to such a lab. So if you are in a bigger city, let's take the example of India. You are in a bigger city which has lots of labs. Maybe this is quick to do. But if you are in a smaller town that does not have a microbiology lab, then you have to transport the sample, and you have RNA, DNA in the wastewater, which also has a lot of other chlorine and many other toxic elements in there. So the nucleic acid will degrade. So then this is time critical. You have to do it. You have to do the sample collection and processing very quickly. So that can add to the cost. So I just mentioned about the labs. So here are some pictures of some of the labs, just to give you an idea of the kind of facilities that are involved in doing such work. But then in resource limited settings, it's not possible that everyone who may be at risk would have access to such facilities. So that's what we wanted to do something about. So one of the things that we tried to do was simplify the sample processing steps. So this is a typical sample processing workflow that I've shown here. So you start from collecting the sample, you do some inactivation to sort of kill the pathogen so that they don't infect whoever is handling the sample. So that can be done with heat or UV treatment. And then you go through a series of steps before you do your DNA measurement using RT-PCR. Sorry, qPCR. If you have to do viral RNA measurement, then you would do qRT-PCR. So we wanted to do something about simplifying this workflow. So that's where we turned to this dipstick-based method of doing it. We had a demo on this yesterday also. These are just photographs of the dipstick. I will just go to this slide. There's a video. So we did some work recently, which is on medRxiv. So if you are interested, you could go read that there. I just have a video that we uploaded as a supplementary material for that paper. So I'll just play the initial part of this, which has a little bit of graphical illustration of how the workflow works. And then I'll skip the variability analysis here.
In this video, we present an overview of the variability analysis study for dipstick-based pathogen detection in wastewater. Wastewater samples are collected in triplicates and heat inactivated to make them safe to handle. Filtration and thermal lysis are performed to release nucleic acid from pathogens present in these samples. Viral RNA is captured using cellulose dipsticks by 3 separate operators. The dipsticks are immersed several times in the tube to bind the nucleic acid to the cellulose. Then the dipstick is dipped in wash buffer to remove cell debris and impurities. Finally, the RNA is eluted by dipping the dipsticks in cDNA master mix. The cDNA is used as template for real-time qPCR, and the CT values for each sample are recorded. To understand the various factors that affect
so I'll skip the variability analysis, but it's as simple as what you just saw. So the sample processing gets cut down into a few minutes step, and there are no instruments needed. In that work, we then went on and did qPCR to do the measurements, but we also work on sensors that can avoid having to do qPCR. So I'll talk a little bit about that, but first let's understand what goes into qPCR. So you collect the samples and then you elute them into this PCR master mix. So it has all these primers and other chemicals that you need to run the PCR reaction. But you also have these probes, the green colored thing that you see here. So these are fluorescent markers. So when your DNA amplifies in this PCR reaction, these probes also get attached to this DNA that gets generated and that amplifies as well. And finally, you then do a fluorescence readout. So you try to estimate the concentration of DNA by doing this fluorescence readout. But then again, you need a fluorescence readout instrument. So this also adds to the cost of doing the test. The probes themselves are also very expensive compared to, so if you just have to do a PCR where you're not doing this fluorescence measurement, those primers are cheaper, but then if you have to add these probes, that adds to the cost as well. So we started looking at electrochemical sensors and thought if we could use electrochemical DNA sensors instead to do this measurement. So in the chart that I've shown on the right, that's a voltammogram for differential pulse voltammetry. I'll not talk about the details of what that is, but the idea is that I have this electrode. So the sensor that I've made, I immobilize these red color strands, which are complementary to the DNA that I want to sense, which may be in my sample. So when I have the red color strand, I see some signal, which is this red curve here. And when I add my sample, if there is DNA that is complementary to what I have on my electrode surface, that will come and bind. So my surface changes, the electrode surface changes. So charge transfer properties of that surface change. As a result of which, if I do the differential pulse voltammetry measurement again, the current should reduce. All right. So this change in current is the sensing mechanism that we use. So this work we started in 2020 when COVID-19 first started. So we did not have a lot of access to many of these things. So we started by thinking of, and we avoid this immobilization of these, what we call functionalization, adding these complementary strands on our electrode surface. And we avoid these qPCR reagents, what redox probe we should use. So these were the questions on our mind back then. I'll just quickly tell you what goes into electrochemical sensing. So it's just a current versus voltage measurement like I showed in the earlier slide. But instead of this, what you would typically do where you have the DUT, two electrodes, and I measure the current and vary the voltage. Instead of that, you vary the voltage across two electrodes, right? But and then you measure the current flowing through the working electrode, but you measure the potential of the working electrode relative to a reference electrode. So the reference electrode is not participating in the reaction. So I wouldn't get a good idea if I just do a two electrode measurement because I'm varying that potential also. So this three electrode measurement is just a better way of doing this voltammetry measurement. So that's maybe all I'll say about it. There's a lot more detail there, which I'll skip. This was how ugly it looked in 2020 when we first started working on this, but we continued improving. So this is the potentials that we started developing. It looks like this now. You will notice in the image on the right that we work with printed circuit boards as electrodes. We had a couple of them at the demo yesterday as well. So we chose printed circuit boards because during 2020 it was not possible for us to get many screen printed electrodes. So this was easily doable. And for redox probe, we thought we'll use methylene blue because we were using methylene blue as a dye in some of the other experiments we were doing. So we thought let's just use methylene blue. So methylene blue has an interesting property. It goes and intercalates with DNA. So it will go and kind of lodge itself somewhere inside the DNA. And methylene blue is also a redox active. So it undergoes a redox reaction, which electrochemical sensing is all about. You measure the current change due to that redox reaction. So then it's also a good chemical to use for this experiment. What we noticed, so in the middle panels, I've just shown what the DPV currents look like, but we can skip that in interest of time. But what I've shown here on the right are photographs of some of these electrodes that we made. And what you'll notice is this green curve is just methylene blue, and then the blue and red curves are methylene blue plus DNA. So comparing only methylene blue to methylene blue and DNA added together, and you allow this methylene blue to bind, and then you do the measurement. So unlike the earlier slide where I said, or even here where we said that if the DNA concentration increases, the current should reduce. Here you see the opposite happening as the DNA concentration increases. So the blue is 5 and red is 10 nanogram per microliters. You see the current actually increase. So this was a bit surprising. So we were thinking what could explain this. We don't, of course, have any functionalization of the electrode. So we realized that this was happening due to adsorption. So you can verify this by doing a cyclic voltammetry measurement instead of differential voltammetry. And then you look for the current versus voltage, but then you look for the exponent. So if the exponent is half, then you know the charge transfer is because limited by diffusion. If it's 1, then you know it's because of adsorption. So whatever species are contributing to the current come and adsorb on the electrode surface. So that was what was happening here. But this was a sensing mechanism that was not explored previously. So we went ahead and did some work here. Another interesting observation we had was, if you see in the plot on the bottom left, there are two different slopes for these curves. So we have DNA concentration on x-axis and the sensor output on the y-axis. The blue colored curve is a much longer DNA fragment. It starts from here and goes all the way till the end. So it's 943 base pairs. So it's a much larger fragment compared to the red one. So the red one is just this 72 base pair portion here in the beginning. So what this told us was that the linearity as well as the sensitivity are better for longer fragments instead of shorter fragments. And we wanted to understand in some more detail. So we did a follow-up study on this where we looked at an organism called 5/6. So it's an enveloped virus similar to SARS-CoV-2, but it doesn't infect humans, it infects algae. So it's easier to handle in the lab. So we started working with that. Sorry, it's not algae, it infects bacteria. So it absolutely does nothing to humans. So it's just easy to handle that. We saw similar observations. So longer fragment, which is 503 base pair, you see this much better linearity and sensitivity compared to 117 base pair. But also you see two different regimes. You see increasing trend in the beginning, and as the DNA concentration increases, you see the current starts to reduce. So there are two different mechanisms at play. The first one, where you see the increase, is explained by adsorption, and the second one is explained by steric hindrance. So there seems to be some concentration dependent kind of thing going on here, which was not studied before. And what we saw is when you add salt, so this was all with pure DNA, purified DNA in the sample. But when you add salt, the adsorption limited sensitivity is compromised. But the steric hindrance still shows you this decreasing trend in current at larger concentrations. So that was another observation which we made that others had not previously talked about. I'll skip this slide. We are now going towards impedance spectroscopy measurements instead of voltammetry, simply because it's more sensitive at some of these lower concentrations where the sensitivity is compromised because of this salt that I was just showing you. So just to give you an idea of what this is, again, we have a 3-electrode system, but we now measure the impedance between the working electrode and the reference electrode instead of measuring the potential between those two. And the electrical equivalent circuit looks like this. So you have a resistor that is the solution resistance, and then you have a charge transfer resistance and a double layer capacitance. So these two are, so this RC which is in parallel, this is the circuit. So it's like the equivalent circuit that is signifying the interactions that happen at the surface. So this is your sensing mechanism really. So most literature talks about RCT, this charge transfer resistance, and they look for changes in RCT as a sensing mechanism. That requires you to do this impedance spectro. So this is a spectroscopy measurement. So I have to do impedance measurement at various frequencies. So if I have to extract this equivalent circuit and get a good measurement of RCT, then I have to do measurements down to extremely small frequencies. So I have to typically start at 0.1 hertz, or I mean, you start at higher and go lower. So you start at 100 kilohertz and go all the way down to 0.1 hertz. But to extract QDL or CDL, you can start at 100 kilohertz and go down only to 10 hertz. So that is, I mean, it may look small, the difference here, but in terms of how much time it takes to do the measurement, it's quite large. So the higher frequency measurement down to 10 hertz, that takes only 15 seconds. But if I have to go down to 0.1 hertz, that takes 10 minutes. And these PCBs have copper tracks under the gold, and there are pinhole defects in the gold. So the longer you keep your electrolyte on the PCB, the higher the chances of copper corroding, which you see here. So this affects the reliability of these electrodes. So we recently did this work where we thought we'll explore the CDL as a sensing mechanism instead of RCT. I'll skip most of this because I want to talk about the infrastructure monitoring, but this is published. So if you're interested, you can have a look at this work. What we show here is that CDL also has a good sensitivity. So here you can see this is DNA extracted from wastewater. So the complementary, you see an increasing trend in the current, but for the non-complementary sample, you don't see that. So it's very selective as well, compared to the earlier methods that I was showing where you don't use functionalized electrodes. And we showed a bunch of selectivity studies in this paper also. There's a lot of variability still, which needs to be addressed. So that is something we are working on. And repeatability across different electrodes, because these electrode surfaces are very rough. So what to do about that? All of those things are things that we are continuing to work on. This is the next part of my talk, which is on infrastructure monitoring. We recently started this company. I'll kind of quickly go past this. Let me come here. So what problem are we trying to solve for infrastructure monitoring? These images, I mean, are they were very widely circulated in media. So I'm sure you recognize some, if not all of these accidents. But the theme is that many infrastructure projects in India do not kind of complete their service life, even before they're recommended, or the life for which they are designed. Much before they attain that life, they have to either undergo major repairs or they undergo catastrophic failure, such as what you see here. So bridges, a lot of bridges collapse in India. I mean, more than 2,000 collapsed in this 40-year period. Uh, what is really concerning is the average failure age of bridges is just 34 years, but the recommended age is 100 years. So imagine you spend all that money to construct a bridge and then it just fails in just 34 years, right? So that is a big problem. Residential building incidents are also increasing. So overall impact on the economy is quite large because of corrosion and other problems that affect the life of infrastructure. So we look at both ways of doing inspection. One is what is called non-destructive testing, and the other is structural health monitoring. So this is just a quick medical analogy to what these two things mean. So non-destructive testing is like a one-time checkup. So you go to a doctor for a regular checkup. Maybe they run some tests and they tell you, oh, maybe there's something concerning, so let's do some more checkups, right? So it's like that. It's not triggered by any particular discomfort or something, but it could be triggered by that, or it could be periodic, right? So it's a diagnostic technology. It will tell you exactly what the problem is, right? You may have to do a lot of tests, but you will eventually figure out the problem. Structural health monitoring is like wearing a smartwatch. You are not wearing it to kind of check every day if you're developing some condition, but over time it has collected a lot of information, so it can start telling you trends that are appearing over time. So it may say, or your blood pressure seems to be increasing in the last 1 year, so maybe there is a problem. So then you cannot diagnose something from this, but then you can go and do non-destructive testing. Right. So then you can go do a check and find out what the problem was, because I'm starting to see something abnormal. Right. So both are needed. The health monitoring system is like an early warning system. The non-destructive testing system is like a diagnostics system. Right. So both are important in any inspection, especially structural inspection framework. So maybe we started late, so I'll take 5 more minutes. Yeah. So there are a lot of different types of non-destructive test techniques that are available for concrete. We can look at these two, which are for which standards are available. The others are also very widely used, but there are no standards for those. The half cell potential measurement is the most widely used technique. It's a probabilistic technique. It doesn't tell you exactly how much corrosion has happened, but it tells you that, oh, there is this much probability that this much corrosion has happened. So it's not a exact measurement. Now, what you have to do there is you have to expose the surface of a rebar. So inside concrete, you have the steel reinforcement. So you have to remove some of the concrete cover and connect one electrode. So this is also an electrochemical measurement. You connect one electrode to the reinforcement and one electrode on the concrete. And then you do a open circuit potential measurement. That's all that it is. But you need a potential stat here as well. So like I said, it's a probabilistic measurement. You have to do some sample preparation. So you have to expose the rebar in order to do this measurement. And it requires calibration because my electrodes are going to age all of these ones which I connect on the outside. So all of this has to be done. Resistivity, we can skip. This also requires similar, is also an electrochemical measurement. When the sensing mechanism relies on the electrochemical changes that happen because of corrosion, but same types of problems is also probabilistic. Pulse steady current is something that was not very widely explored for rebar, so I'll talk more about what we did there. It is an expensive technology, but we have done something to kind of address that part of it. So whatever I've shown in red, we have done something about that. But let me spend a little bit of time talking about this electrochemical impedance method, which was not really widely explored in concrete inspection, and this is inspired by MEMS. So before doing all of this work, I used to do MEMS. So I thought I can show you how some of those MEMS ideas translated here. So what is this sacrificial anode? So like I said, corrosion is an electrochemical reaction. So steel will rust over time and form oxide, right, which is the more stable form as the structure ages. So if I want to protect my steel from rusting, I can insert another material which is more electrochemically active. So that may rust instead. So that is zinc. So zinc is more electrochemically active than steel. So in the construction, you can use the sacrificial anode. So they are called sacrificial because that rusts but protects the steel from rusting. So in that sense, it's sacrificial, and it's the anode in the electrochemical reaction. So that's why it's called a sacrificial anode. All it is a zinc disc that is tied to the reinforcement with galvanized iron wires, right? So that you establish an electrical connection. The zinc corrodes, the steel does not corrode. And you have to put an electrolyte around it, which is a conductive mortar. So the conductive mortar works as the electrolyte. So what we did was, so there's no monitoring technology. So sacrificial anodes are used not in reinforced concrete infrastructure, but if you go to a shipping yard where they're working on ships in a dry dock, you will see on the underside, the hull of the ship, you will see lot of small pieces that are just bolted down. Those are all sacrificial anodes. And if you ever get a chance to look at a marine bridge, a bridge in a sea, you will see lot of these in the piers that go into the salt water. Right. So these are very widely used there. They could be used in a building like this also. But there's no monitoring technology. So once you put in these anodes, you don't know how long they are going to last. So then you do some periodic replacement. So every 3 years, maybe you replace the anodes on a ship, etc. That's how it's done. So we thought we'll monitor this. So our sensing mechanism was directly go here. We connect a piezoelectric transducer to the zinc anode. So this is just a piezoelectric wafer transducer, the kind of thing which we use in our ultrasonic testing. We attach it to the zinc disc. So now it looks like a resonator, MEMS resonator. I have a transducer on top and I have this large mass on the bottom. And if I perform a electromechanical impedance spectroscopy, because this is not electrochemical, I see this typical resonance susceptance spectra that you get for MEMS resonators. And when this anode corrodes, because of change in mass and geometry of the structure, the resonance frequency shifts. So that is the sensing mechanism that we use. There are two types of resonances, series and parallel. I'll maybe skip the details in interest of time, but we track the series resonance, which in this case corresponds to this breathing mode of vibration. So there's a circular disc which expands in and out. So that's the resonant mode that we look at. I'll skip this, all this maths. All that I'll say is, if you look at how the mass change should happen because of corrosion, which is this first equation, Faraday's law, and just look at over time. So this is just time over which we are applying current to induce corrosion. But you can think of the x-axis as age. So over time, the change in mass should happen linearly if my corrosion current is constant, so my frequency shift should be linear. But in the experiment, we see this extremely non-linear shape, which are these squares that I've shown here. So we did some work and figured out that there is something going on with the delamination of the corrosion byproduct. So zinc will form zinc oxide. So when that delaminates from zinc, this interesting non-linearity appears. So we did a lot of work to understand that. What is good about this is this also works when this resonator is surrounded by solid concrete, right? Because MEMS typically, if you have liquid or something surrounding it, the quality factors really drop and the sensing is affected. Here, because anyway, my structure is very large, so the sensing mechanism can still be used. It is still non-linear, but then we did a lot of simulations and again, we can model and understand the non-linearity. So what we learned was we can use this method, so we'll see where that goes. But eventually, this technology also we would like to take to the startup. This needs a little bit of more work, but the other one which we are going to commercialize. So I have a last few slides left. Is this pulsed eddy current? So I started by saying pulsed eddy current was not explored for concrete, reinforced concrete, because it is expensive and well, there were no other. I mean, there are products, but they're just very expensive products, so not as widely used. The idea is simple. You have coils. So this sample is just the rebar. You can imagine it that way. So I have coils inducing eddy currents on the sample. So I have changing current in my coils, which will induce the magnetic field will induce current in my rebar. And because of the time varying current, the eddy current will also be time varying. So eddy current will induce its own magnetic field, which I detect with a magnetic field sensor. So that's all it is in terms of the idea. So this work first started in 2018 as a course project, and then a lot of students continued working. So Durgesh, who did that smart anode work, also worked on this in his PhD. We continued doing a lot more work. So this we've launched as a startup now. I mean, just in the last month, December 24. So this is my last video. So I'll just show you, because this we are taking to the startup. I don't want to share too many details, but if you have questions, I'm happy to answer those. So it's just a quick video. There's no audio here, just showing glimpses of the technology. It's a handheld device. There are, like I said, a lot of products that are out there, but their accuracy is not very good, and the cost is very high. This is usually used for monitoring pipelines. So pipelines, your sensor and the pipeline surface are very close to each other. But in concrete, the sensor is on the surface of a wall and rebar is very far away inside the concrete. So that is what causes some challenges which we have overcome. And as you can see in the video, the instrument is very small. We actually did a live demo of this at last year's AppsCon. And almost exactly 1 year ago, we were here at Inventive, just before last year's AppsCon showing this. So a lot of work has happened on this in the last 1 year. So I mean, we could not bring it to this year's AppsCon, but if you are interested, I can talk to you about it later. So with that, let me conclude and thank all the people who have done all this work. So Shruti is here along with Avni. She did all the wastewater processing work that I talked about in the beginning. Durgesh and other students and staff worked on some of the structural health monitoring work that I showed. A lot of collaborators to thank for all the work we've done. I am also leading this Vadhwani Electronics Lab at IIT Bombay, where I mean, a lot of staff and resources from the lab have also helped this work significantly. And my co-founders for the company, Ankur is a staff at the Vadhwani Electronics Lab, who is also a co-founder for the company. Pavitra and I were colleagues earlier at Analog Devices, and Shavik Banerjee is my collaborator with whom we started doing some of this work. And then our funding agencies, WRCB, GCRF, and DST, who supported the environmental monitoring work, and SERB and our industrial partners who have supported the infrastructure monitoring. So thank you. This was my last slide.